Okay, so this poll everywhere question is obviously relates to yesterday's class where we talked about Alzheimer's disease. And if you, as clearly you all remember, which is great, it's the one of the factors in Alzheimer's disease is the loss of this cholinergic projection from the basal forebrain through the cortex. So the thing which, and it's fantastic that you all remembered that. And so the thing to sort of keep in your heads is that I could, with this exact same set of options for answers, like, you know, what would you pick if the question had been about ALS? Correct. And although we haven't got there yet, if we were talking about addiction, you'd be picking A. And if we were talking about schizophrenia, you'd be picking D. So all of these things sort of like, you know, like if you just have them in isolation, they're easier to remember. And it's sort of just trying to sort of keep some level of awareness of all of these different pathways, because there's so many different pathways that we'll be talking about, you know, like through this section. And so it's like trying to keep them straight is going to be like the challenge. So today we'll be continuing to talk about neurodegenerative disorders, and this time we'll be talking about Parkinson's disease and Huntington's disease. And both of these have specific features that make them classed as movement disorders. And it's because they, you know, like they both have sort of damage to sort of, you know, like widespread areas of the brain, but they have sort of a particular focus of damage in particular areas that are involved in movement. So for Parkinson's disease, we're going to lose the dopaminergic neurons in the substantia nigra. And in Huntington's disease, we're going to lose medium spiny neurons in the striatum. So how does that relate to movement? So there's this structure, which is the basal ganglia. And the particularly we have the striatum and this projection from the substantia nigra. And the striatum itself is formed of like two structures called the putamen and the caudate nucleus. And interestingly, only primates actually have that subdivision. Sort of all other mammals just have a single kind of glob together type thing, which is probably related to the fact that primates have more fine motor skills. So these structures are important in the release of motor programs. So if you remember the stages of movement that we talked about, gosh, I can't remember, a couple of weeks ago, we have these sort of three major stages of movement in order to sort of do some voluntary motion. So we have a planning stage, a programming stage, and an execution stage. And so last week we talked about ALS, where we are losing the motor neurons, right? The upper and lower motor neurons. So that is obviously a failure of execution. So you can't execute any kind of motor action because, you know, like you literally don't have any motor neurons in order to cause the muscles to contract. Motor planning is obviously something that goes on in many cases in primarily in the cortex. So because it's like related to, you know, like I'm going to lift up this, but I'm not going to ask a question just yet. So that's motor planning. However, if you think about all of the different muscles I'm going to use in order to go like that, you know, like it's not just sort of like contract muscle A. There's a whole bunch of different muscles that all have to contract with different levels of force. And other muscles have to be relaxed in order to, so I'm not fighting myself. So those patterns of muscle activation are motor programs. And, you know, we have huge numbers of, you know, like motor programs that we've sort of learned. And in order to initiate any kind of motor act, we have to release those specific motor programs. How does that relate to Huntington's disease and Parkinson's disease? So one of the main, so a major role of the basal ganglia is the selecting and releasing of motor programs. And this involves sort of two things which are called the direct and the indirect pathway. But the sort of greatly simplified version of this is that, so okay, we have the cortex to create our motor program, and then it passes through the striatum, and then will go out to the motor cortex, where, you know, like the motor cortex obviously houses the upper motor neurons, which then will go down to activate the lower motor neurons, which will then activate the muscles. So the output is always going to go through the motor cortex. But the bit of selecting which motor programs to use comes through the striatum, and that is strongly influenced by these dopaminergic projections from the substantia nigra. So we have this, and so this dopaminergic input modulates and helps select the motor program, and it can be excitatory, i.e. triggering the activation of this motor program, or it can be inhibitory, i.e. suppressing the activation of a particular motor program. So if you think about it, you always, you have this huge array of potential motor programs. You don't want them all going off at the same time, otherwise you'd be literally like flailing around like crazy. And so you have to have both the selection side and the suppression side. And so that's what the direct and indirect pathways are doing. And both of those, you actually use dopamine as a neurotransmitter, because depending on which dopamine receptor you're using, it can be inhibitory or stimulatory. If you have insufficient ability to trigger motor programs, then you'll have hesitancy and difficulty performing tasks, or even like complete rigidity. And so that's fundamentally what's occurring in Parkinson's disease. If you have difficulty in suppressing motor programs, you'll have uncontrolled motions, and that is termed chorea, and with a chorea, and rather than the country, and that causes sort of like flailing sort of arm movements and things like that, because they're not being suppressed, and that's a hallmark feature of Huntington's disease. So it's the loss of these direct versus indirect pathways in these two movement disorders that gives rise to the sort of these classic motor features of the diseases. So to talk about Parkinson's first, we have these sort of very classic motor symptoms that you can sort of, like really easily see in a patient. So, like the classic signs are going to be sort of like if you're sort of talking about sort of gross motor features, as the disease progresses, people, like tend to sort of stoop forward. They tend to have very sort of shuffly walking, because they have difficulty sort of, like initiating the normal motor movements for walking. Typically there'll be rigidity and trembling of the head and sort of hands and so on. Often there's like going to be a resting tremor where they're sort of like kind of like this. They'll also tend to have intention tremors, which is even if they don't have much of a resting tremor, so I'm wanting to pick up that, they'll sort of like have the pronounced tremor before they can actually initiate the motor movement. So these are all down to this loss of this dopaminergic pathway. There are also, and generally all of their movements will be very slow and sort of deliberate seeming, and that's called bradykinesia. It's the slow movement. They also have some other motor features that you wouldn't probably think of, which is that their faces will tend to be rather mask-like, because they have difficulty in all of those little motor movements that create our expressions. And there's less eye blinking, a very soft voice. And interestingly, if they sort of like, you do a sort of handwriting, look at their handwriting, it tends to get really, really tiny. So it's both slow and painstaking and gets really, really small. And that's again because of the difficulty of sort of making these sort of movements, and so it just kind of gets minimized. And in addition to this, there are often large numbers of non-motor features that sort of develop as the disease progresses. And so these are in many cases, are going to be down to sort of damage of other brain regions. But in particular, things like there are many autonomic features that Parkinson's patients will develop. And sort of like one of the classics, for example, is going to be orthostatic hypertension, like where, like they might have high blood pressure sort of when they're sitting down, but when they stand up, they're unable to sort of adapt appropriately. And so then they'll often, fall over just from, like having a sudden abrupt drop in their blood pressure. Yes. Yeah. Yes. In that pathway, yes. So those are the G protein coupled receptors. So that is a good question. So slow is relative to the ligand-gated ion channels. So those ones, can give like their maximum response in sort of like 5 to 10 milliseconds. If you're signaling through the G protein coupled receptors, the quickest responses you can get are going to be on the order of more like 20 milliseconds. So it's still not super slow. And the reason that it doesn't matter that it's slow is the fact we're actually modulating the passage of information. So the actual, this is where, like I could easily do an entire couple of classes probably on the brain circuitry that's involved in this. So this is very simplified. But basically what's happening is that you have a direct fast neurotransmission that is going to essentially flow all the way through from cortex to motor cortex down to down the spinal cord to the muscles. So that's all going to be fast. However, the motor programming portion of that is actually modulating those fast circuits. So they're suppressing or stimulating them. And so that, because it's modulating, it doesn't have to be quite so quick, because it's sort of like, like putting we're adding, like a multiplier, or we're, like adding a divider to sort of try and speed up information flow or slow it down, kind of. So that's why it doesn't really matter that it's a little bit slower than if it were like just ligand-gated ion channels. Yes. So for today's class, we're not talking about norepinephrine and epinephrine. They're going to be the same kinds of speeds as dopamine. But they're not. They're also signaling through G protein coupled receptors. So I've already told you, like what is this, like that we've lost this population of neurons, well, lost its input from the substantia nigra using dopamine. And this is just showing the pathological findings in patients who have Parkinson's disease. And so this is a section through, like kind of the brainstem. And here you can see these kind of like dark marks. And this is actually the substantia nigra. That's why it has that name, because they're sort of like heavily pigmented cells. So you can actually see them. And so in a Parkinson's patient, you can see that they've almost totally disappeared. And if you look at a sort of microscopic level, which is shown below, this is the sort of the same cells. You can see them, like obviously it's a different way of staining. But you can see, like lots of cells here, and here you have hardly any. So we're not just losing, like neurons, like we might in, say, Alzheimer's disease. We're losing a specific population of neurons. And that's why we're specifically losing this dopaminergic projection. But just like Alzheimer's disease, there is a protein aggregate that we can find in these cells, and these are called Lewy bodies. And these are composed of primarily alpha-synuclein. So these are dopaminergic neurons. So they're neurons which use, for when they're talking to other neurons, they use dopamine. They don't necessarily themselves have dopamine receptors, but their projections are going to be dopaminergic. So the cells that are dying have these sort of protein aggregates, these Lewy bodies. And in some cases, they will also have some of these neurofibrillary tangles using the tau protein. So just like yesterday, our big questions are going to be sort of, like, why are these neurons dying? And there is very definitely a good number of different genes which carry mutations which will lead to Parkinson's disease. In general, if you have these mutations, like, you will develop Parkinson's disease. So it's not like a susceptibility gene. These are typically causal genes. And typically, in those cases, Parkinson's will also develop, earlier. Whereas in idiopathic Parkinson's disease, like, where it just kind of randomly occurs, the sort of typical age of onset will be sort of round about 60, something like that. However, there are also a bunch of environmental factors that have been linked to Parkinson's disease, because the genetically based Parkinson's is actually pretty rare. However, these sort of sporadic idiopathic Parkinson's disease is, is the bulk of the Parkinson's population. So what are the risk factors? Well, one of them is actually living in sort of healthy, like, countryside air. So rural populations are actually more at risk from Parkinson's disease than urban populations. So, typically we think about sort of cities as being sort of creating greater health risks, but specifically for Parkinson's, it's actually the opposite way around. You're less at risk in a city. And this is basically because there's seem to be environmental factors that play into Parkinson's disease risk. And so in particular, obviously, people who live in a rural place will, frequently have, well water, for example. And so there might be sort of certain metal contaminants which increase the risk of Parkinson's disease. There are also pesticides that will increase the risk of Parkinson's disease. So this is thought to be the reason why Parkinson's is more common in rural populations. Depends. If it's a true, like, reverse osmosis one, yes. So, but like the sort of little, like, was that? Yes, those ones probably less. They'll reduce some of those things, but not as effectively as the full reverse osmosis, like, system. So. No, actually, that's a good question. No, so in like, it doesn't have any increased sort of so like we talked yesterday about how generally the brain will shrink, and so it'll be subject to all of that, but no quicker than the rest of the brain. So in general, that doesn't seem to be a factor. However, it has this like, sensitivities to certain toxins. So if you're exposed to those things, then like, even if you have a really low level of exposure, then it'll still shrink quicker than other areas. And if you have a high level, then it'll shrink quickly, or not shrink, but the neurons, the specific neurons, will die. Yes. Which bit? So there's kind of these genes, which you don't have to generally remember, though alpha-synuclein might be a good one since there's the one in the Lewy bodies. Those genes lead to loss, they still lead to the same features. So they still lead to the loss of the substantia nigra dopaminergic neurons. But in other patients, they don't have those genetic mutations, but they still develop Parkinson's disease. So if you harbor those mutations, then you get an earlier onset Parkinson's disease, and it's pretty much guaranteed. So it's the same neurons dying in both cases. So because of this link to sort of suspected contaminants, it's led to this neurotoxicity model for Parkinson's disease. And some of this actually comes from people who managed to give themselves Parkinson's disease. So there was an outbreak in, I think it was California in the 80s, where suddenly they had a bunch of 20-year-olds with Parkinson's disease coming in. And it turned out that they were trying to synthesize a drug, like of abuse, and had a contaminant in it that generated this protein MPTP. And basically what it did was basically very rapidly, like in the space of a couple of days, killed off their substantia nigra dopaminergic neurons. And so suddenly you had someone who a week ago was a healthy 20-year-old suddenly becomes a full-on sort of advanced Parkinson's patient. And so that is, like, was obviously terrible for them. They did sort of eventually figure that out. Then there was another outbreak in Canada from the exact same synthesis about 10 years later. And so, like, it does sort of pop up every now and then. And what this drug does is it can pass across the blood-brain barrier. And in glial cells, it gets converted to this MPP+. And that is a substrate for the dopamine transporter, which means what kind of neurons will take it up? Dopaminergic neurons. So the dopaminergic neurons take it up, and inside the dopaminergic neurons, it will basically destroy the mitochondria. And so those neurons will then die. So it's a very sinisterly targeted chemical, because it will, like it just through random happenstance, it is able to specifically target those one neurons. Yes. I can't remember. I believe it was oral, but I can't remember. So there are other drugs which seem to have some similar features. And so this is, for example, rotenone is a pesticide. And it's ironically a pesticide used heavily in organic farming. And it is, I guess it counts as organic because it's a naturally occurring pesticide. But it's still toxic. And so it's lipophilic, and so it can also cross the blood-brain barrier, and it also seems to accumulate in dopaminergic neurons, and also acts as a mitochondrial toxin. So we have these drugs that are specifically targeting the dopaminergic neurons, and so because we're losing those through toxicity, we're generating features of Parkinsonism. Many of the genes that are, cause this almost guaranteed Parkinsonism are also associated with mitochondrial function. And so, as we've talked about, like if you have any kind of issue with ATP supplies, then you're going to end up with cell death. So that's how these environmental contaminants seem to be able to sort of induce the specific loss of dopaminergic neurons. And one other feature, and it gets its own special slide, is that there are drugs, clinical drugs, that will also lead to Parkinson's features. And the good thing about these is at least these aren't toxins. So this isn't something that causes irreversible loss of these neurons. These are drugs that are basically, they're called neuroleptic drugs. They're acting in opposition to dopaminergic signaling. So when they're present in the brain, they're suppressing dopaminergic signaling. And if they do it to too strong an extent, then they'll induce Parkinson-like features. You can take them away, and then the patient does not have those features anymore. And they're called neuroleptic drugs because these are sort of drugs that are used for treatment of schizophrenia and so on. So it's not a toxicity, it's just a pharmacological action that is reversible. And so if you're taking one of these, if a patient is taking one of these neuroleptic drugs, then the development of Parkinson-like symptoms is something you have to watch for, and sort of to try and do a sort of risk-benefit analysis for those patients to sort of see, okay, we don't want them to be suffering from severe Parkinson's, but we also don't want them to have their other symptoms as well. So just quickly, how would we go about helping people with Parkinson's disease? Sadly, we don't have any way of regenerating the lost neurons. And it's not clear whether or not there's, we can even sort of halt the progression of the disease. But we can do a lot of sort of relatively short-term symptomatic relief. And fundamentally, this all comes down to supplementation of the dopamine. So we can't just give them dopamine because, for one, it would cause a major reaction in their blood vessels, and two, it doesn't actually cross the blood-brain barrier. So we use levodopa. And levodopa is a precursor for dopamine. And it's usually combined with something that prevents its breakdown outside of the brain. And we'll talk about this a lot more in pharmacology, but there are complications with the use of these agents, just because over time, the patients will develop these sort of on and off periods where they're responding or not responding to the drug, even though they're taking it. And so that leads to sort of more issues with sort of, again, medication management, and sort of, do you use other agents to try and boost monoamine signaling, like inhibitors of the monoamine oxidase, things like that. There are also direct dopaminergic agonists as an alternative to sort of supplementation of the dopamine. And if you're managing sort of Parkinson's patients, there's this whole bunch of sort of clinical pharmacology that is related to sort of trying to help with their symptoms. So just as a slightly different approach, I want you to sort of, when you're talking together, I want you to try and think of the most confusing aspect of this last section, or the hardest bit to understand, and talk about that. Dopamine.
That's how I spell it myself. You can get to it. Same one, we just see a different size.
Okay. So we're going to call on someone to explain or at least talk about whatever they found most difficult. Who's the lucky person? Levodopa, but yes, or L-dopa. Yes. So that's a good summary and a good question. And so basically, it won't specifically, like it won't target itself, but it will target itself in the same way actually as those toxins do, because the dopaminergic neurons will take up the dopamine, and they'll just sort of be able to, basically package more dopamine into their synaptic vesicles, and so they'll be in that way they'll be releasing more dopamine than they would without this extra supplementation. So it's basically strengthening the dopaminergic signaling in much the same way as the Alzheimer's medications are strengthening the cholinergic signaling. So we're just trying to, it's just essentially trying to sort of help the surviving neurons to signal stronger. Okay, so we will now talk a little bit about Huntington's disease. And so this is the guy who discovered this. And I think it's kind of funny that he only ever wrote 2 scientific papers in his entire career. And this one was when he was 22 and had just graduated med school. So, however, he didn't, but this was the defining description of Huntington's disease. And it was heavily reliant on the fact that his was a family practice. And so he also had his grandfather and father's medical records for the community. And because Huntington's is a genetic disorder, the fact that he had this sort of multi-generational study in a small isolated geographical community, so Long Island, New York, that was what allowed him to sort of see these features of what it became Huntington's disease. And I know this is kind of long wordy slide, but it's like kind of neat that it describes all of the features of Huntington's disease, like pretty much as we view it today. So I will read this, but so I wish to draw your attention to a form of the disease which exists, so far as I know, almost exclusively on the east end of Long Island. It's peculiar in itself and seems to obey certain fixed laws. In the first place, let me remark that chorea, as it is commonly known to the profession, and the description of which I've already given, is of exceedingly rare occurrence there. The hereditary chorea is confined to certain, unfortunately a few families, and has been transmitted to them, an heirloom from generations way back in the dim past. It is spoken of by those in whose veins the seeds of the disease are known to exist with a kind of horror, and not at all alluded to except through dire necessity when it's called that disorder. It's generally attended by all the symptoms of common chorea, only in an aggravated degree, hardly ever manifesting itself until adult or middle life, and then coming on gradually but surely, increasing by degrees and often occupying years in its development, until the hapless sufferer is but a quivering wreck of his former self. He says there are three peculiarities in the disease: its hereditary nature, a tendency to insanity and suicide, and it's manifesting itself as a grave disease only in adult life. And of its hereditary nature, when either or both of the parents have shown manifestations of the disease, and more especially when these manifestations have been of a serious nature, one or more of the offspring almost invariably suffer from the disease if they live to adult age. But if by any chance these children go through life without it, the thread is broken, and the grandchildren and great-grandchildren of the original Shakers may rest assured they're free from disease. So this is this fantastic description of a hereditary dominant disease. And as he described, it's typically the onset of symptoms is typically in the sort of age range of 30 to 50. And it has this hallmark symptom of the chorea, which is these uncontrolled movements of the arms, legs, heads, face, upper body, and so on. It also has strong features of dementia. So there's a decline in thinking and reasoning. Unlike in Alzheimer's disease, there's declarative memory is actually pretty spared. It's obviously autosomal dominant genetic disorder. And once the symptoms manifest, then there's about a life expectancy of another sort of 15 years or so. So as an autosomal dominant disease, as you'd expect, there is a, clear, there's sort of very clear genetics. So it's just classic autosomal dominant pattern. If both of your parents have it, you will have it. If one of your parents have it, you have a 50% chance of having it. What's that? Well, so that's, once you develop the symptoms, it's that 15 to 20 year period. So it will be, so it is lethal, but yes, exactly. So there's no symptoms until you're past, typical childbearing age. So it doesn't, there's no negative selection against it. So that's how, I guess, as a disease, it sort of has progressed for, remained for so long. So, no, it wouldn't. That's the point, because the homozygous. So, but in this case, you can have two bad alleles because it affects you in middle age. Yeah, the lifespan is reduced, but no embryonic lethality. So it's kind of an interesting genetic disorder in that sense too. So and obviously Huntington himself wasn't able to identify the gene, since they didn't even know what a gene was at that point, but it has been identified and named after him. So it's the Huntington gene. And the actual mutation that's found in these patients is actually a triplet repeat that is in the middle of the coding sequence. And so in healthy individuals, sort of non-carriers, typically there's going to be 10 to 26 of these repeats. In carrying individuals, there's going to be 37 to I think, 80 repeats. And you don't need to know the sort of number of repeats. But so you get a big expansion of this triplet repeat. And the size of the expansion correlates with the severity of the symptoms. Okay, great. So, so it's specifically this expansion of the triplet repeats that gives rise to the symptoms. And if you look here, this is the sort of proteome of the Huntington, And this will hopefully explain why I'm not going to try and really explain what the protein does, because it's actually not very clear. It does a lot of different factors, and in general, it's thought to be particularly involved in organization of signaling processes within the cell in some kind of vague way. Why it particularly kills these medium spiny neurons rather than other neurons is not clear in the mutated form. But there are, like many different areas where the protein function, like, so if you, for example, take a cell line and, knock in this mutation, you can find dozens of problems with signaling, with sort of the mitochondria, with the cytoskeleton, with intracellular trafficking. There are many different things that aren't working quite right. And so it's basically at that point, people are kind of throwing their hands up in the air and saying, okay, we don't know which of these is actually particularly significant. Yes, you had a question? ROS, reactive oxygen species. Species, yeah. So we know that genetically, we know exactly what the problem is, but cell biologically, we haven't the faintest idea, because it could be sort of pretty much any of these things. No, this is a copied figure. So this is the ones that the people writing that review article wanted us to sort of focus on. So I'm more showing this as, like my sort of get out of jail free card, because I, like can't tell you exactly which of these is going to be the most important. So you need to be aware that it has widespread cell biological effects, and that's kind of it as far as the like detailed cell biology of this disorder. But what it does lead to is you sort of get relatively early loss of those medium spiny neurons, but in later stages, you get something that looks much more like Alzheimer's disease, where you actually have, really pretty widespread neuronal atrophy. So this will probably remind you of that picture I showed of the actual brain specimen I showed yesterday for Alzheimer's, like where it's all shrunken and shriveled. This is obviously in a living patient, but the same kind of thing can be seen here. You can see these sort of folds are much more, like obvious than they would be in a healthy individual, and the ventricles are greatly enlarged, which is what the arrows are pointing to. So there's this widespread shrinking of the brain, which is, like obviously what's leading to the sort of widespread dementia in these patients. Yes. So first it kills off the medium spiny neurons in the striatum, and then yes, then the sort of effects spread further and further throughout the brain. And so in late stages, it is looking more like Alzheimer's, though with less problems with memory. So we have this particular pattern of disease progression. So in stage 1, the patient will be, like starting to exhibit some of these sort of, like arm movements and so on. But other than that, will be sort of, fully functional. And in stage 2, there'll be, like the disease will have progressed, so it's starting to have an impact on cognitive function. In stage 3, which is the sort of 5 to 16 kind of range, there'll be the patient will be basically, like incapable of doing most things. And then, out in the sort of 10 to 20 year range, they sort of need sort of, full-time, nursing care and so on. What could we do to help these patients? So we have the cardinal symptom is that motor issue, and it's the opposite motor issue to what you would see with Parkinson's patients. And so the symptomatic relief because again, we only have symptomatic treatments. It's going to be the opposite to what you do in Parkinson's patients. So in Parkinson's patients, we're trying to boost the dopamine system. So in Huntington's patients, we're trying to block the dopaminergic signaling. And so this can be neuroleptic drugs, these sort of dopamine antagonists, or it can be things that just, for example, prevent the filling of the vesicles with dopamine, which is what tetrabenazine does. So VMAT is vesicular monoamine transporter. There are also these other sort of psychogenic type symptoms. So there's like irritability and in some cases severe anger and threatening. And so those you treat with antipsychotic medications. For other sort of milder psychological symptoms, you might treat with an SSRI. These patients will often have features of anxiety and depression and insomnia, and so those would be just individually treated as well. So like whether it's like sleep aids or antidepressants or anxiolytic drugs and so on. But like fundamentally, it's like again going to be just one of these progressive neurodegenerative conditions that like there is pretty much nothing that can be done other than to sort of make the early stages as troubling as possible. Okay, and the last one of these, at least for today. And there will be no questions on people. Just not to interrupt. Actually, you get to. But I think Very last thing, which is just about the quiz. So that's next Tuesday, right? And so Dr. Phipps has agreed. So he said that it would be sort of 5 out of 6 classes. And so what we've agreed is that there will actually be 6 questions from all 6 classes. However, you only need 5 right, if you see what I mean.
Can you answer all 6?
You can answer all 6.
Okay.
So you can study all. I would prefer you to study all 6 classes. But yes, but you only need to. You can answer all 6 questions. You only need to answer 5 of them correctly. If that's clear.
So do you get extra points?
You do not get extra points.
Oh.
Because they get very hard to keep track of.
Yes, but then you
It does.
really don't know.
So the ones that he's posted on Canvas, I think it's from the. Was it epilepsy was the last one with the exam, so it's one after was it Friday? But anyway, 6 classes. Yes. It will be multi-group. And if we enter all 6, take away points from us and we get back. No. So, yeah, if you get 5 right, you get 6 right, you still get.